there's probably others in this room who could think of many more examples than what's shown here, but met with a number of our experts across our institutions, and just a few examples of various things that are happening. So in our Heart Center, there's research going on whether repeat sequences in the genome are predictive of outcomes from cardiac interventions. In the fetal and neonatal area, rapid next generation sequencing that's used to determine whether the condition of a child or whether they would be responsive to a particular drug in order to hopefully get them out of our NICU faster. Obviously in cancer, we do tumor sequencing to try to find the drugs that might be most effective for that particular patient with the lowest number of side effects. And then in our brain and spine area, we have many examples of where this is being used. And one example that was shared with me around a patient that had a very long-term condition, they finally were able to do some sequencing and figure out that a simple $10 a month generic drug could actually solve the problem, but it had never been tried for this patient. And so overall, what we're talking about when we're talking about precision medicine is how do we improve management and informed decision-making? How do we more speedily solve uncommon problems? How do we reduce complications and time on treatment for patients? And how do we improve quality of life? And so, it's really about this whole idea of healthcare value that Primary Children's is so focused on, increasing treatment efficacy and reducing healthcare costs. So what we're hoping to do is, although all of those things are happening somewhat independently right now, is there an opportunity through this exploration to build somewhat of a precision medicine center? And it may or may not be a physical location, but more of a wraparound of services. That includes leadership, it includes focused faculty and all the different staff that are needed to support precision medicine. It includes all of the resources that are necessary, a biobank, tapping into the Utah Population Database, our data warehouses on both the university and the Intermountain side, and our various analytics expertise to try to bring about greater ability to reach patients faster, and we're really interested at that point of care. How do you actually, down at this lower part, how do we actually build into our electronic medical record the opportunity to say, we've done some genome sequencing on this patient and these drugs would be most effective, and then we're going to track their clinical outcomes and we're going to use that knowledge to pass forward to the next generation, both here and nationally. So as part of that exploration, we've invited Dr. O'Day to come out and join us, give grand rounds, and then we're going to have a series of meetings with her over the next couple of days because she really is an expert on precision medicine, as you will see. So I'm going to do a quick introduction, and her bio is huge, so I'm going to cut it down a little bit. But Dr. O'Day is the CEO and the founder of Olaris Therapeutics, a precision medicine company that uses pioneering metabolics platform and proprietary machine learning algorithms to fundamentally improve how disease is diagnosed and treated. Dr. O'Day is also a member of the World Economic Forum's Global Future Council on Biotechnology, and she serves on the advisory board for the Precision Medicine Initiative and was an invited speaker at the annual meeting in Davos. She was also a nominated changemaker and invited to attend the first United States of Women Summit convened by the White House in 2016. She participated in Vice President Joe Biden's Cancer Moonshot Summit to discuss collaborative ways to end cancer as we know it. In addition to that, Dr. O'Day is the founder of Lizard Fashion, an apparel company that uses fashion to promote science, and Project Spark, a nonprofit that recycles electronics and uses the resale of parts to build computer centers and orphanages throughout the world. Additionally, Dr. O'Day is adjunct faculty at Boston College teaching metabolism and entrepreneurship. Dr. O'Day received her PhD at Harvard University, where she was a National Science Foundation Fellow. She's one of 11 American scientists awarded a Winston Churchill Fellowship and received a Master's of Philosophy and Chemistry from the University of Cambridge. In full disclosure, Dr. O'Day and I are actually longtime friends. We met in undergrad at Boston College. We were both studying science. She was studying biochemistry, I was studying biology. And we were both awarded the same scholarship. It's the Beckman Scholarship. So for those of you who don't know, Arnold Beckman was the inventor of the first commercially available pH meter. He was also the inventor of what's the word for it? It's the oxygen analyzers for NICU incubators that keep babies from going blind. So he and his wife started a scholarship fund to encourage undergraduates to pursue a career in research. I was studying cytokinesis in sea urchin eggs. Liz was studying something that's way over my head that I don't understand and probably led to what she's actually doing today. So one of us actually parlayed that scholarship into a very successful career in research, and one of us did not. So I will take this opportunity to formally apologize to the Beckman Foundation for my failures in that area. However, you will see very quickly that Liz is doing more than enough within her career for both of us, so kind of all works out for Boston College. So please join me in welcoming Dr. Liz O'Day.
All right. Thank you for that generous introduction and not too embarrassing. I appreciate it. As Erin said, we've known each other for years. And I think if both of us could go back and tell our sort of 18, 19-year-old selves that we'd both be standing here today on International Women's Day, no less, we wouldn't have believed it. But I'm very proud of all you have accomplished, independent of going into research or not. So thank you for having me, and thank you all for coming today. I am really excited to have the opportunity to meet with a lot of you over the next couple of days to learn about your clinical work and to tell you about our research and to discuss ways that we may be able to work together. As sort of a foundation for those conversations, I'm going to give a quick introduction into precision medicine and tell you a little bit about what we do. So even though it's early, we're going to start today with a little audience participation. I need your help in telling me which one of these pieces of artwork is better. So take a moment and tell me, we're going to do this by a show of hands, how many people in this room prefer the three musicians by Pablo Picasso? Raise them high and proud. Got it. Okay. And how many of you guys are Monet fans and prefer the water lilies? Great. I myself am a water lilies person. But is one of us right or better? Is one of these artworks categorically better than the other? No. That's because art is often driven by, personal preference, and art is subjective. In medicine today, actually, there are a lot of similarities to art. Sir William Osler, the father of modern medicine, once said, if it would not be about the variability among individuals, medicine could well be a science and not an art. Because of the differences between patients, today, many clinicians have to act like artists, but instead of sort of blending different watercolors on a canvas, they're stringing together different treatment protocols. And there are some amazing clinician artists out there, and their patient success stories turn out to be masterpieces. But that's not always the case. Not every patient turns out to be a Mona Lisa. Precision medicine aims to sort of even the playing field and for all patients so that we move medicine away from an art into more of a data-driven science. So, what is precision medicine? Precision medicine uses a patient's molecular makeup to guide health decisions. Biomarkers, including genes, proteins, metabolites, microbiome byproducts, epigenetic markers, and more, have all been identified that can differentiate subpopulations to their susceptibility to a disease, prognosis of disease, and response to treatment. Basically, what that means is that there are biomarkers that make you, you, and me. Those same biomarkers explain why you are susceptible to certain diseases and myself others. Further, biomarkers can explain that if you and I were to have the same disease and to be treated with the exact same drug, for one of us, we might have a successful outcome, but that may not be the case for the other. So, precision medicine aims to use biomarkers to get towards better treatment decisions. Why do we need precision medicine? Well, the answer is sort of economics, honestly. The global healthcare spend continues to rise at unsustainable rates. For full disclosure, I poached this slide from another talk I give on value-based healthcare, but I think it drives home the point that we need to sort of reevaluate and become much more efficient in our healthcare systems. So shown here, let's see if I can do this, is the health-adjusted life expectancy, or the HALI, on the left-hand side or on the y-axis, where we take the life expectancy in any country, so the developing countries are shown in the dark blue, and the developing emerging countries are shown in the light blue, and you take the life expectancy and subtract years where people aren't living at optimal health. And then we correlate this against the health expenditure. And ideally, what you want to see is that as a country spends more on its on the health of its population, that you improve the health-adjusted life expectancy, or the HALI. So this would sort of be your ideal path. What you don't want to see is that despite spending more and more, thousands and thousands and thousands of more dollars, you're actually not making any improvement to this improved quality of life. And so what we can see here is that we are continuing to spend thousands upon thousands of dollars, and we're not making that much improvement. And unfortunately, it's estimated that more than 1/3 of this spend is on wasted treatments, either drugs or procedures that simply don't work. So we need to be a lot more efficient in the sort of the healthcare and the medicine that we offer patients. I'll have someone in the room take a guess. Where do you think the U.S. is on this graph? Yeah, we're the worst. And so, you can see that there's a real need for us to sort of reevaluate how we approach healthcare. And I'm happy to chat more about value-based healthcare towards the end of this if anyone has any questions. The need for precision medicine is also highlighted when we look at drug responder rates. And I think that this sort of striking image from this 2015 Nature paper captures that. Shown here are the top 10 U.S. grossing drugs, and the number of patients needed to treat, or the NNT, ranges between 4 and 24 for just one patient to benefit from that drug. That means the majority of patients being treated with these drugs are not being helped. And in fact, sometimes, because of adverse effects, they could actually be harmed. So the question I have to you is, why is it acceptable to administer drugs to patient populations when we know they only work in maybe 1/4 of those patients? And further, why are we spending hundreds of thousands of dollars on those drugs when we know that they don't work? Wouldn't it be smarter to use the patient of information to drive better healthcare decisions? Well, I think that's possible. So one of the things we say at Olaris, my company, is that, imagine this. Imagine you or someone you love got the results of a biopsy back, and it showed that it was cancer. This can be a terrifying moment for an average person and also for clinicians as well, because we know a lot of the nuances of how this works and doesn't work. So you get this result and you say, okay, I want to start fighting right away. But how do you fight? How do you pick which treatment will work for you? Well, any clinician, probably most of you in this room, will agree with me that we have access now to better cancer treatments than ever before, but still we have not figured out which patient will do best on which drug. This leads to this sort of trial and error driven medicine, which can be both frustrating and terrifying. But I will tell you, it doesn't have to be this way. There is technology, stuff that I'll tell you about my company, but other companies that exist already. Ta-da! Should I keep? Okay. Okay. That was like a big reveal. We're going to get into Olaris technology next. So as I was saying, it doesn't have to be this way. Technology already exists to get the right drug to the right patient, to bypass this sort of guesswork so that people can immediately get on the road to recovery. And it is with this in mind that I started Olaris. So Olaris, as Erin alluded to, is a company I started 3 years ago as a spin out of my PhD. We use an NMR, nuclear magnetic resonance-based metabolite profiling platform, as well as machine learning to, as naive or idealistic as this sounds, fundamentally change how diseases are treated and diagnosed. So the goal of our company is twofold. We aim to uncover novel therapeutic targets, which we think could be more relevant to a disease state, as well as to develop patient stratification tools to help optimize therapies, ultimately getting the right drug to the right patient. So I'll take you through some of that. So first, you guys might be asking, why metabolomics? If you take anything away from today's talk, it is this. We need to sort of break free from the gene-centric tunnel vision of genes will be the answer for dictating which drug will work for which patient. Genomics is great, but it only provides copy number information. You actually need to look downstream of the genome to include all relevant biomarkers that can inform health decisions. So In our case, we've decided to look at metabolites because metabolites are sort of the downstream summation of everything that happens in a cell. So while DNA and RNA tell you what could happen and proteins tell you who's making it happen, the metabolites are a functional readout of the cell telling you what is actually happening in the cell. They really relay information about all sort of the upstream genomics as well as inputs from the environment. And Olaris' technology can detect and quantify nearly the entire human endogenous metabolome. So there are 3,000 endogenous metabolites that swim around in you and I, and it is the rate and synthesis of breakdown of those molecules that, again, makes you, you, and me. There are also microbiome byproducts, which Olaris' technology can measure, and drug breakdowns. Not everything that we ingest in our bodies is good for us, and it will be broken down into different pathways, into different people. And so being able to detect and quantify that can provide very powerful information. So our technology, or the ability to sort of map the full metabolome, just like a genotype, we refer to this as the metabotype. And so the first case that I want to show you about is actually in our drug development programs, where we were using this technology to develop precision medicine for triple-negative breast cancer. So as I'm sure there's many of you in this room, breast cancer, many in this room know breast cancer is a very heterogeneous disease. It can be classified in multiple ways, but one of the most standard ways is based on the cell surface receptors, or based on the expression of cell surface receptors, including the estrogen receptor, progesterone receptor, or HER2. And then there's a group of breast cancers that don't overexpress any of these markers, and they are called triple-negative breast cancer. They usually account for about 10 to 20% of all breast cancers. These sort of are the worst ones. I kind of liken them to sort of the junk drawer in your kitchen. Unfortunately, these patients don't fit into any of the other molecular subtypes, and as a result of this, there are no positive diagnostic markers for them, nor are there tailored therapies, so the prognosis is honestly the worst. So there's a real high medical need to uncover diagnostics to identify these patients, as well as tailored therapies to improve their outcomes. So using our technology, that's exactly what we set out to do. And so we made use of a cell line model of triple-negative breast cancer to start. So this was how this was derived. We took normal breast tissue, or this was done in Bob Weinberg's lab at MIT, took normal breast tissue and cultured it in different medium and gave rise to these two different cell types known as BPLAR and HMLAR. These are isogenic cell lines, but the BPLAR cells, as you can see here, have a high tumor initiating capacity. So if you take just 100 of these cells and inject them into the mammary fat pad of a mouse, they will develop tumors. Whereas the HMLAR cells, you actually need 10 to the 6 cells to get any sort of palpable tumor in a mouse. So these BPLAR cells are often considered models of sort of cancer stem cells, if you believe in that word, or tumor initiating cells. And the tumors that they give rise to resemble triple-negative breast cancer. So using this as a model system, we used our NMR method to say what are the metabolites that are expressed in TNBC cells, and what are the metabolites that are expressed in HMLAR cells, and can we use the metabolism as a readout to figure out what's going on in this BPLAR cell line. And for those of you that are not familiar with NMR spectroscopy, this is what a spectra looks like. It's called a heteronuclear spectrum quantum correlation, or an HSQC, where you get a peak for every carbon molecule attached to a unique proton. So protons on the Y, or on the X, and carbon is on the Y. And so basically every peak here is a different metabolite. And what you see is that about 80% of the metabolites between the BPLAR and the HMLAR cells are conserved, which kind of makes sense. These are both highly proliferative cancer cells. But there were a handful of metabolites that were only expressed in the triple-negative BPLAR cell line, and using our Olaris proprietary library, which has over 9,000 reference peaks, we were able to figure out what are those metabolites that are unique to TNBC and then probe their biology. And so what we found is the most significantly upregulated metabolite was this molecule called N-acetylnurmic acid, or NuA5AC. So NuA5AC is the most abundant member of the salicylic acid family of sugars. Salicylic acids are the terminating residues of many cell surface glycoproteins. Olaris developed proprietary software to sort of figure out the pathways of which salicylic acid is made. And what happens is that first glucose goes through the hexosamine pathway before then it is converted by this enzyme called N-acetylnurmic acid phosphatase into an N-acetylnurmic acid, which gets shuttled into the nucleus, where this enzyme CMAS activates it into the CMP intermediate, which then gets shuttled to the Golgi, where these enzymes called transferases can then attach this sugar to cell surface glycoproteins. So from our technology, we figured out that this pathway was sort of going gangbusters in triple-negative breast cancer. And we wanted to see then, okay, if this pathway is truly upregulated is the end result to increased salicylic acid or salation on the cell surface of triple negative breast cancer cells? And the answer is yes. We proved this using rhodamine labeled wheat germ agglutinin. So wheat germ agglutinin, it binds to anything that contains this sugar moiety, and we added a rhodamine level to it. So you can see that there was a lot more fluorescence on the triple negative breast cancer cells than the HMLER cells, suggesting an increase in salivation. Further, triple negative breast cancer cells are known to be highly invasive. And in the slide introducing triple negative breast cancer, high rates of metastasis are often described with this disease. And one of the in vitro models for metastasis are these invasion assays where you grow cells in a dual chamber. They have a matrigel separating the top and bottom layers. If the cells are metastatic, they can sort of burrow through that matrigel and form colonies on the other side. So you can see the TNBC cells can do this quite well. You stain it with crystal violet, whereas the non-TNBC cells can't really do that. If you treat these BPLAR triple negative breast cancer cells with neuraminidase, an enzyme that will remove this sugar from cell surface glycoproteins, what happens is quite striking. Now BPLAR cells are no longer able to invade, suggesting that this pathway leading to the production of increased salicylated glycoproteins is important for the malignancy of triple negative breast cancer. So then we wanted to say, okay, so we know that this pathway is important for driving the malignant behavior of TNBC. Is there a therapeutic target in here that we can then manipulate that could lead to a tailored therapy? We decided to focus on CMAS. So this enzyme right here sort of performs the critical step in the biosynthetic pathway. And so we decided to say, okay, what's CMAS doing in TNBC? And we made use of the Cancer Genome Atlas. For those of you probably all still are familiar with it, the TCGA contains DNA and RNA levels of about 10,000 different cancer patients. There's maybe 2,000 of them are breast cancer. And so we decided to look at what is the CMAS, DNA, and RNA expression level in the TNBC subpopulation within the TCGA. We did this by first measuring the levels of estrogen, progesterone, and HER2 throughout the entire breast cancer population in the TGCA to identify what would be considered a putative triple negative breast cancer subpopulation. And we found that indeed there was about 15% of all breast cancers in the TGCA could be labeled as triple negative breast cancer using this methodology, which correlates to what you see in the population. And while in TNBC, you see that these markers are downregulated, you see the opposite with CMAS, where CMAS copy number variation, so DNA can be twofold to sometimes even eightfold higher in triple negative breast cancer patients, and the mRNA is also increased. I would say these are very modest increase, so only 1.2-fold as well as 1.5-fold, although, incredibly significant, but this also highlights the need to look past the genome, because as I will show you going on, we're pretty confident that CMAS is going to be an exciting target for triple negative breast cancer, but if you just ever focused on genomics, you might not have got here. So we then sort of decided to probe, okay, if we think CMAS is a target, let's figure out what it's doing and see if we can come up with an intervention for it. And so what we found is that CMAS is actually sort of the master regulator of invasion. So again, we took these triple negative breast cancer cells, did these invasion assays, overexpressed or transfected an siRNA control, and you can see BPLAR cells can still invade. But now when you knock down CMAS, they are not able to invade whatsoever. And the reciprocal is actually true. So where you take non-triple negative, non-aggressive cells, overexpress an empty plasmid, these cells are still non-invasive. But when you force the expression of CMAS, now they become wildly invasive. And to us, this suggests that CMAS is sort of this master regulator and could represent, a potential therapeutic target. This was further validated when we took this into animal models. So we made BPLAR, shRNA, or SH, constitutive CMAS knockdown cells. So these cells constitutively express an shRNA to knock down CMAS as well as control cells, and we injected them into the mammary fat pad of mice. And what we found is while all of the mice that were injected with the control cells developed large primary tumors as well as mets to the lung and the liver, none of the mice that were injected with the knockdown cells ever developed a tumor. And we injected 500,000 cells here. And if you remember from my first slide, only 100 of these cells is required to form a tumor. So this suggests that CMAS really is a potential, novel, important therapeutic target for triple negative breast cancer. And so with that, we then set about creating a drug program around this. So we cloned CMOS, we expressed it recombinantly, went through all of those processes, and we have now identified a lead compound with nanomolar inhibition and anti-cancer activity. So Olaris is currently developing a precision medicine for triple-negative breast cancer, and just to remind you of how we did this is that we started with the metabolites. We took metabolites in TNBC cells and non-TNBC cells and looked at the differences. We then linked those to metabolic pathways to figure out who was up and down regulated, and does that point to a potential therapeutic target. We made use of the genome editing tools at the time, which were, siRNAs were all in vogue then. Now we could probably use CRISPR or something like that to turn off that target and see, does it have the phenotype that we hope for? And if the answer is yes, well, then you can start the path of drug discovery. And that's where we are now, where we're now evaluating CMOS expression in triple-negative breast cancer biopsies, as well as exploring or furthering our small molecule drug program. So this is our lead compound. It's C105, aptly named because it was the 105th compound that we tested. And you can see here that it blocks colony formation in this sort of, again, in vitro assay of a tumor initiation, where you take single colonies, and if the cells are tumorigenic or able to form a tumor, they can form, colonies quite well, which are sort of supposed to be representative of a tumor. This is a very poor inhibitor, C9, which kind of decreases that, but C105 completely prevents tumor formation. And so we are now using this C105 to do rounds of medchem, and in 10 years, and if we're really successful, we might have what could be sort of the best-in-class and first-in-class treatment for triple-negative breast cancer. And so we are actually doing this for a number of programs. So TNBC is our lead program, but we're also tackling mutant KRAS-driven pancreatic cancer. So pancreatic cancer, as I'm sure we all know in this room, is terrible. Nearly everybody that gets it will unfortunately die from it. And here's a case that's really frustrating because we know the genomics. Over 90% of pancreatic tumors have a mutation in KRAS. So what we did here is we took whole tumors that express either wild type or the mutant KRAS and looked how that mutation changes the downstream metabolome. And I will say, much like our TNBC story, we found metabolites uniquely expressed in the mutant KRAS story. Perhaps those will be diagnostics that could lead to earlier detection, but for sure they also point to potential therapeutic pathways. And we're doing this for a whole series of other diseases, including CDKL5, which is a neurological disorder, several hepatic diseases, and I'm excited we just started a program also in Parkinson's disease. The other half of the company focuses on developing what we call biomarkers of response. And here, I like both sides, but I think there's a lot of potential room for impact here. And with the biomarkers of response, what we intend to sort of tackle is the treatment problem. So 1 in 8 women will get breast cancer in their life. It's a striking statistic. And despite greater understanding of the molecular drivers that are responsible for breast cancer, it's still really difficult for clinicians to pick the best treatment. Shown here are some of the top sort of 4 hormone receptor or estrogen receptor breast cancer drugs. And these drugs, the responder rates are shown here. And, in the best case scenario, ribociclib works in about 50% of the patients. So it's the same as a flip of the coin, where the majority of chemo drugs, like their response rates hover in around the 20% range, meaning most of the women that are going on these drugs are not being helped. And in fact, a lot of these drugs have adverse or serious side effects. Further, because these drugs aren't cheap, this exposes families to what we call financial toxicity. One of the number one drivers of a middle-class family going bankrupt is a primary cancer diagnosis, or cancer diagnosis in the primary family, which I just think is such a shame. We have to be able to do better. So what leads to sort of this treatment variability is drug resistance. So the majority of breast cancer patients or any cancer patient will at some point during their treatment experience drug resistance. This could be intrinsic resistance, which exists before a patient is ever given a drug, or acquired resistance, that while the patient is on the drug, the drug no longer has the intended effect. And the molecular mechanisms driving drug resistance are complex and honestly not well resolved. Drug resistance can be due to decreased drug uptake, prodrug into activation, increase of drug efflux pumps, or a whole series of different molecular pathways. And so this is sort of what gives rise to this trial and error treatment paradigm that we're currently stuck in, is because we can't tell which patients are resistant to which drug. We just kind of have to put them on it, see what happens, and then adjust accordingly. I think that by using biomarkers and going beyond genomic biomarkers, we might be able to do better here. And so a couple of examples that I just wanted to highlight is for Herceptin. So HER2-positive breast cancer patients, there's about 30% of them that are actually intrinsically resistant to these targeted therapies. So Herceptin was a game changer and has definitely improved HER2-positive breast cancer outcomes, but it doesn't work for everybody. And there's technology that will let us kind of figure that out. The same thing for acquired resistance. Tamoxifen, again, just staying in the breast cancer space, is one of the most widely prescribed ER-positive breast cancer drugs, but almost half of the patients during treatment will develop acquired resistance with this. And we should be monitoring and detecting who these patients are so that we can optimize their treatments accordingly. So how Olaris intends to do this, again, and now instead of trying to uncover healthy versus disease, we're using our metabolite platform to differentiate responders versus resistant populations. And again, just like before, while earlier studies have been focusing on covering genetic biomarkers, we believe that adding metabolites or additional biomarkers will provide a more functional readout and maybe be able for us to intervene quicker. So using our metabolite platform and machine learning, we identify what we call biomarkers of response that where we can detect metabolite signatures, and these can be endogenous metabolites, microbiome byproducts, as well as drug breakdown products. We take all this together, we use the AI and the machine learning to see, is there a pattern of metabolites that can correlate with clinical outcomes? And I'll share with you some of our first results. So our first program is again, in breast cancer, looking at CDK4 and CDK6 inhibitors. So these are a relatively new FDA-approved drug. I think they have been out there for about 2 years now, and they've become sort of one of the frontline therapies for ER-positive breast cancer. But unfortunately, due to intrinsic drug resistance, there's a small subset of women that actually, when you put them on this drug, their tumors grow faster. So they are, according to RESIST guidelines, they have what's called progressive disease, that patients who, within 2 months of treatment, will have at least a 20% increase in the sum of the diameters of their tumors. Whereas other patients, in that same time point, we look for what's called a partial response, where there's at least a 30% decrease in the sum of their tumors. And so in a small subset of only 6 patients, which we are now expanding to over 200, we were able to identify a pattern of metabolites that could differentiate the women with progressive disease and those who had at least a partial response. And so our assay, which, as I said, we're expanding to further cohorts, could ultimately one day be used to screen patients so that before they ever get administered this drug, we see which group do they likely fall in. And this would hopefully allow them to sort of get on the road to recovery a lot quicker. And I should say these drugs aren't cheap either, so it would also save both them healthcare system as well as the families a lot of money. And so what's sort of sad, I guess, is that nearly every drug and every disease has these sort of responsive and resistant patient profiles. And so we are, in fact, conducting BOR studies in a lot of different areas. We have a tamoxifen study and a aromatase inhibitor study, also in ER-positive breast cancer. We're looking at how at taxane-induced neuropathy, also in breast cancer, to see if there might be a biomarker signature that could detect when for which patients neuropathy is a real risk factor. We're looking at satutinib in renal cell carcinoma. Satroplatin is a really interesting example. For those of you who might know it, satroplatin was being developed for prostate cancer, but it no longer exists because it failed to meet its phase 2 or phase 3 clinical endpoints. So millions of dollars and at least 10 years went into the development of this drug, but because it didn't, work in the large prostate cancer population, it's dead. We think that we were able to identify, there were certain patients that it worked in, and so we think that we'll be able to identify a metabolic signature to identify the patients that it worked in so that you might resurrect this drug and get it to people that it can actually benefit. So we're doing this in a whole other slew of cancers. One of the really hot ones is with these all new, everybody wants an IO drug right now. They're all the rage. Immunotherapies are great. For some people, you'll see their tumor-stricken bodies, you'll see all those tumors disappear, but actually IO drugs are twice as likely to kill you as they are to cure you. And so figuring out which group that you likely fall in, I think is incredibly important and worth looking at. And going beyond cancer, we're also now doing studies in psoriasis, heart disease transplantation, and we just started one in autism, trying to sort of see if we can help add a little bit of quantification to a very subjective disease. And so I guess one of the reasons I'm here these next couple of days is to kind of talk to you and see if there, are ways, or let you know that we are certainly interested in collaborating with clinicians and healthcare institutions to identify BORs in therapeutic areas of need. And this is where we need to sort of learn from you, or where are the most pressing issues? Where would a biomarker in your clinical work be really helpful? Where would one of these help you improve the treatment for your patients? Because as naive and cliche as this might sound, I think that together we can really identify signatures to tailor treatments, improve outcomes, and save lives. And so how you can start to think about integrating precision medicine into your own practice is one of the things at the forum we like to say is that we encourage all clinicians to think of every patient as an N=1 clinical trial. So for every patient, collect some biomarkers, whether that's, I would encourage a baseline as well as biomarkers throughout their treatment so that we can start to map those biomarkers to clinical outcomes and throw those outcomes into EHRs, get broad patient consent so that they can contribute to these research projects, and also making sure that we're safeguarding patient privacy is incredibly important parts of this. But if we start to do this for every patient, we can then sort of aggregate all of these results, and then we'll be able to figure out sort of what was the data that led to that Mona Lisa patient, that patient that had this particular signature. They survived not because of some subjective whatever. They survived because you got them the right treatment, and there are biomarkers that will be able for us to sort of go back and figure that out. And in fact, I know that Erin mentioned you guys are already sort of making headway and getting into the precision medicine space. I think you guys are doing incredible work, so great job. I think the biobank that you have been telling me about will be a really rich set of data that could really transform the lives of the people here. In 2016, I was invited, or I went to Davos to the World Economic Forum with all the, the glitterati and the big leaders of the world to give a talk about precision medicine. And this was the title of my talk. It was that biomarkers are the DeLorean of personalized medicine. So do we have any Back to the Future fans in the room? Okay. So the DeLorean is, the time machine. It's what lets Marty McFly and Doc Brown travel between past, present, and future. Biomarkers are what let us do that for patients. Biomarkers of the past are already helping us transform treatment for present-day patients. Let's take CML, for example. Prior to 2001, less than 1 in 3 CML patients survived past 5 years. But then, because of genomic sequencing and other efforts, the Philadelphia chromosome was discovered, and the BCR fusion protein and Gleevec, or imatinib, was developed to particularly target these patients. Now, CML has a survival rate, or a 5-year survival rate of over 90%, which is incredibly dramatic. And so if we start sort of collecting and investing in biomarkers now, we'll be able to do the same thing for future patients. And I think this will go beyond just cancer. There terrifying statistics that it's like less than, I think, only 1 out of every 9 children responds to the ADHD drug they're on. Fewer than 40% of depression patients respond to the depression drug that they've been put on. And so there's a real need in all these different therapeutic areas to start tailoring treatments to that individual so that we can improve outcomes. In fact, it's estimated that 70% of decisions that are made by doctors like you in this room use a biomarker. But what's striking, or on the opposite end, is that less than 2% of the $2 trillion spent annually on healthcare flows to diagnostic providers. That means that diagnostic companies are not being compensated for sort of the value that they're providing. And as a result, I will say that the diagnostic business landscape or commercial opportunity is very limited because while investors see a big ROI on developing a drug, you don't get that same sort of value in diagnostics. And so this is where we really need to develop new public and private partnerships to invest in biomarkers. So this is some of the work that I do on the World Economic Forum, is try to set up, interesting collaborations where it brings in both, private industry like Olaris and clinicians and healthcare providers so that we can sort of do something meaningful to make impact. And I would say that I think here you guys really have that opportunity. I think that based on, the Intermountain healthcare system, from what I've seen so far, you guys could really become a leader in biomarker ID. You've got a tremendous population genetics that's working for you. It's less noisy than a lot of the other areas that, we do studies with. You already have a pretty impressive EHR system put into place, and the ability to sort of track these families over long periods of time is something that other areas don't have. So I think that, in the future, it'll be very interesting, or, we'll see what happens, but you guys certainly have the potential to identify biomarkers that can distinguish healthy from diseased, as well as those that are responsive to resistant and those that are resistant to different therapies. And so why we all do this is actually to give kids the best treatments. So neuroblastoma is a rare form of cancer that develops from nerve tissue. And there's about 650 cases that are reported each year in the U.S., and my older brother, Rob, was one of those cases. That's me when I was a child. Rob is doing fine and has survived, and he runs marathons, Ironman, and he's great now, so that's wonderful. But for 2 years, my family lived in Boston's Children's Hospital, and I saw toxin after toxin being pumped into my brother. He turned into like a skeleton, and we weren't sure what was going to happen. Thankfully it worked with a whole lot of like hope and prayer, but I think that by investing in biomarkers, we can actually do better. And so I just, remind everybody, if you were a patient or a patient's loved one, wouldn't you value a test to let them pick the right treatment? And with that, I will pause and take any questions.
Questions?
Thanks for a great talk. So I get how precision medicine will improve outcomes. I don't get how it saves money. You suggested, well, we'll put them on the right drug instead of the wrong drug. But that only works if the right drug is cheaper or they don't get a drug at all. So in the breast cancer illustrations that you gave, resistance, okay, well, we'll put them on a different drug. But how does that actually save any money?
Well, so because of the trial and error treatment, and patients, and you probably can add more clarity to this, often get put on multiple different drugs, right? And so each drug costs money, each drug has its own complications, and time is money as well. And so by optimizing the treatment, by getting on the right drug right away, you're sort of streamlining that to, again, help improve them as quick as possible. And yeah, maybe that you're still paying $150,000 for drug A versus the drug B, but if drug A works for you, all the downstream complications that would come from being on a drug that wasn't working for you are eliminated. And diabetes is another great example that we do work with in China. Diabetes patients get on 8 drugs or something, and it seems that the modus operandi there is that you go on metformin. Does metformin reduce your HbA1c levels? If not, you just add another drug, and then you add another drug, and you add another drug. That is not an effective way to treat patients, and it's just building up sort of this drug wastage problem. So that's maybe sort of how you're getting at it. I would say, then putting on my value-based healthcare hat, is that we actually need to change drug pricing and get around this, right? So you can't charge $150,000 for a drug that only works for 20% of the patients unless you have a companion diagnostic with it, and to get sort of moving towards value-based healthcare. So, yeah.
Hi, Dr. Rudy. That's a great talk. So, as you're probably maybe aware, that there was 2 papers that came out last week in Nature about the largest sequencing efforts in pediatric cancer, and one of the big conclusions was how different the driver mutations in pediatrics are than adults. And what you presented on the metabolites is really interesting. I'm just wondering if have you done anything and looked at any of the differences in adult diseases versus more pediatric diseases?
The short answer is no, but we want to. And that's also, again, highlighting the point of needing to kind of break free of this genome-centric view of that, the genes will be the answer, because in any average patient, I think there's somewhere in that paper and other papers 10 to 12 mutations identified, and it's very hard to know which one is the oncogenic driver. And there's all kinds of evidence that almost 50% of genetic mutations identified are sort of a false positive, that they have nothing to do with that patient's cancer. So we're certainly interested in looking in that in the pediatric population. Also, just to add on that, there's a lot of ethnic differences that also arise. So if we look at CYP proteins and cytochrome P450s that express in your liver, there's all kinds of SNPs, single nucleotide polymorphisms, that vary among different ethnicities, which make you metabolize drugs very differently if you are of a Caucasian background than of a Middle Eastern background or an Asian background. And so we're really going to need to sort of tailor these drugs and dose these drugs to the right aged individual and the right ethnic background as well.
Thanks again for coming. It's a wonderful talk. My question is I saw a lot of data about individual cell lines. Have you done anything to look at the influence of the tumor microenvironment on the metabolomics that could potentially impact some of these therapeutic predictive models that you're looking at?
Yeah, so the tumor microenvironment is, complex, and I think that's also why a lot of drugs don't work, because you design it for one area and then you're throwing it into the tumor micro niche, if you want, and you're having to respond to all of that. So we approach this in two different ways. So when we're trying to develop targeted therapies, we actually kind of really do take a cell-based approach and one of the reasons we looked at cancer stem cells, because if you believe the cancer stem cell hypothesis, these are the cells that are responsible for forming a tumor. So we wanted to say, what is sort of driving cancer formation in a cancer stem cell population? And that's how we came up with our CMOS target. As our inhibitors go through clinical development, we are collecting biomarkers to determine CMOS target engagement within the tumor microenvironment. So we're collecting biomarkers in our animal models to see, like, these mice who we saw their tumors shrink, what were the biomarker signatures there? And does that correlate to, additional non-human animal models? And can we even then bring that into the clinical studies? So it's a little bit of both, you know what I mean? And that's when I have to explain this to my investors, sometimes they don't actually get it. But like what you see inside a cell is not necessarily what's going to be circulated throughout the entire system, because there's a lot of different forces coming together. So yeah.
Thanks for a great talk. This is related to Paul's question about drug pricing. Biomarker pricing is also a problem. Novel biomarkers that we try to use, we cannot either get approved or they're so expensive that patients can't afford them. Can you comment on that?
Which ones are expensive?
Almost all of the new ones.
Like PDL1, is that what you're thinking, or?
I'm not sure what PDL1
Oh,
is.
so I think it,
no,
yeah.
no. So I will give you a specific example. I'm a gastroenterologist, and I try to order fecal calprotectin, and it's over $600. And it's frequently considered to be experimental, so it won't be covered. Or a family is on a high deductible plan, and I have to justify that cost.
I hear you, and I feel this is a problem that we need to tackle, and where we need to bring all the different stakeholders together. The doctors and explaining to the insurance companies or the payers why this test is of value and how it would improve my treatment. So usually we hear the other side of the argument that diagnostics are sort of reimbursed at sort of the rate for the benchtop chemistry required to run that test. So like where a drug, you can charge $150,000 and CMS will reimburse you for that. For diagnostics that still require a lot of heavy lifting in terms of R&D to uncover, maybe best case scenario, you get a couple $100, which for like a company trying to commercialize that and get investors creates a very difficult situation. So what we are trying to do, both at Olaris and my work on the forum, is to engage payers right away and just to say, like, these biomarkers are adding a lot of value. They will, if you used them routinely, they could save you this amount of money. So like if we look at the PDL1 is what I asked you about, that's used for these IO drugs, Keytruda for non-small cell lung carcinoma. So for non-small cell lung carcinoma, Merck has developed this drug, Keytruda. It's an immuno-oncology drug, and if you express high levels of PDL1, then you can get the drug. There's a little murkiness, like whether that is the best biomarker and, all this sort of gray area, and it needs to sort of be clinically validated. We are trying to sort of get our test that would figure out, like, are you the people that are going to die or live on an IO drug? Reimbursed at a much higher rate, because ultimately we're telling the insurance companies that, like, if we can tell you that this patient is going to die on this, isn't that worth a couple thousand or whatever it is? And so kind of doing the math to drive these decisions. I'm not sure I'm totally answering your question, because I think it's a complicated one. I think we need to invest in biomarkers. We need them clinically validated, and that's where we need clinician support so that we can then go to the payers and say, hey, this needs to be reimbursed at this rate or to be completely covered or whatever it is, so that you guys can start to integrate precision medicine into your practice. So I think dialogue needs to happen in the short answer there, but we're still working on it.
Thank you, Dr. O'Day. I have a question related to Dr. Guthrie's. That is, turnaround time is often a major issue with diagnostics, as is quality control. How would your business model accommodate, especially the turnaround time issue? And I think that's especially important in metabolomics.
Yeah, so our business model, to be quite frank, and I can feel my investors telling me to stop talking, is invent, make, sell, right? So we're in the invent stage where we're still even trying to uncover what are the relevant biomarkers, and that requires time. So our NMR experiments, like each one takes about an hour. That's not something that we would ever expect a doctor to wait on. Right now, we want to uncover what is the biomarker or the panel of biomarkers that's then important, and then we can make that in the second stage of our business company into some rapidly deployable test. Think of some dipstick or some ELISA assay or something like that. So that's where we're progressing. But the point that I'm trying to make now is that we are still in the phase where we need to get the biomarkers. We need to collect the right data, again, going beyond genomics and sort of put critical thinking around it to figure out what are the things that we need that would help empower our health decisions. But we're not there yet, so yeah.
No? Erin, any final words? Do you guys always talk this fast between the two?
Yeah, I know. You should have seen us together. Yeah.
I went to Boston University. I don't know if they taught us that there. It's been a pleasure. Thank you. And thank you all for coming.
Thank you, guys. Sorry, I know I'm